Okay, so now we're going to get into DNA replication and how DNA actually makes two DNA strands from one, which is the basic of DNA replication. So we know it's a semi-conservative method, so we know that you have two strands of DNA in here, and in order to make two molecules of DNA, it replicates one strand, separates, and then a new strand comes in and attaches. Semi-conservative method. And we also know, or hopefully you realize, it is all due to a whole bunch of enzymes. And we're going to look at the specific enzymes and keep track of the enzymes involved and make sure you understand what each one does. So I'm going to erase this a little and get started on, as at first, looking at the DNA strand in a large view and how it looks when it's replicating in a large manner. So this is one side of DNA. This is the other side of DNA. And what I'm drawing with these circles is these are what's known as replication bubbles. And this is actually where replication is occurring, at these sites. And notice that there's more than one along the DNA strand. The fact that you don't just start at one end and move all the way along to the end, because that would take way too long. So there's multiple sites of replication. And at each one of these sites, it's occurring in that direction and that direction and that direction and that direction, that direction and that direction. So therefore, it can occur much faster. Now, to start off with, the first enzyme that DNA deals with your lovely double-stranded molecule like this, is an enzyme known as helicase. Helicase is an enzyme that breaks the bonds between the nitrogen bases. So there would be a helicase here, 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 and here. And it creates what's known as a replication fork. And this is going to be moving this way, that way. Helicase can move in all directions. And it doesn't matter, it'll just attach in one location. So if it attached here, it would start moving this direction and create replication. Now, where it actually breaks it apart, you have your DNA strand, and it separates it, and I didn't change color for some reason, forgot. And you have what's known as your replication fork. So it looks like the fork in the road. And at this is where helicase is coming in and breaking these bonds. Now when this happens, you now have two strands with nitrogen bases that aren't paired. And that is where replication is actually going to occur. And that's where the next enzyme, known as polymerase, comes in, DNA polymerase to be precise. So DNA polymerase is going to attach to this and bring in the next nucleotide. So it'll say bring in, because this is a C, always going to bond with a G. This is a T, going to bring in an A. This is an A, going to bring in a T. And same thing over here. You have the complementary. So now you see this piece is the same as this piece. So you see why the complementary base pairing is so important, is because it then makes sure that you have the same exact DNA molecule when you replicate. Now the only tricky part about DNA polymerase is it can only attach in the 5 to 3 direction. So when it looks at a DNA molecule, if it's the 5' end of the actual nucleotide, so here's the nucleotide, and that's the 5' end, 3' end of the nucleotide, it can attach a nucleotide over this end. It can only attach a nucleotide over this end. It's kind of crazy, I know, but so it will only grow in a 5 to 3 manner. Now, if you notice a slight problem with this, is that your two strands are anti-parallel. So you have, I'm going to change color for emphasis over here. Say this is a 3' end, 5' end, 3' end, 5' end, because they're anti-parallel. Notice the anti-parallel nature. This new strand is 3' to 5'. And this new strand, 5, 3. Now, this one, because DNA polymerase goes in a 5 to 3, is being built this direction. This one is being built in this direction. Now, this one looks like it'll have a hum-dandy time because DNA polymerase, I'll change my little, will just be able to easily stay connected to the strand, and as helicase comes through and separates here, it'll just keep moving down and keep adding the new strand. Only problem over here is it can't go this direction, so it's going against the grain, basically. And what's going to happen here is it's going to do little segments. So this strand over here is known as the leading strand. It is a continuous strand. US. Over here is known as the lagging strand. Now the lagging strand has to be built in segments. So it's going to look like, if we have our old strand is this green, and then what's going to end up happening is because I need to go back to green, you have this part was broken off, and this was left empty, so this got made. And then I'm going to erase a little. This part was broken off, and so polymerase came in and built this direction. And then our last little bit was broken off, not necessarily actually broken off, but and then you end up with little fragments because they're all being made in this little direction. So these are what's known as Okazaki fragments. And this is where our third enzyme comes in. And our third very important enzyme is ligase. Ligase will come in and connect these. So ligase will attach here and come in and go, ooh, we can bond those. Ooh, we can bond those. So they make that special bond. It's only one little bond between nucleotides, but it's a very important bond. So then you'll end up with a complete DNA molecule, whereas the other one is going to be complete no matter what. So if you look at this whole process, you start with, let me erase a little over here. You start with a DNA molecule that it has been opened. I'm going to draw it really big. Opened by helicase. So helicase has come in here, and we're going to draw helicase in green like we did before. Helicase is right here, breaking those bonds. Then we're going to make this 3', 5', 3', 5'. So replication is occurring in this direction. And so this new strand you have to look at and say, okay, it's being made 5 to 3 towards the fork. So therefore, this is going to be our leading strand. And this one, because it's going to have to be made away from in order to become 5 to 3, has to be our lagging strand. And that's where polymerase comes in and continually goes this way, and we'll go backwards this way, and then again backwards, and that's where your ligase will come in. And if this made a strand here and here, this will connect that little fragment, and then you end up with your 2 strands of DNA, one with the old and one new strand. And that is your semi-conservative method that makes everything work out perfectly because you end up with 2 identical DNA molecules. So I hope you remember these 3 enzymes, the very important enzymes, helicase, polymerase, and ligase, and realize that as this is occurring, it is in a bubble. So realize because they're going in opposite directions, the leading and lagging strand are just in looking at the replication fork, because this could be over here, the leading, because going this direction, then over here it's going the opposite direction. So this same strand of DNA is known as the lagging strand. Now, it looks confusing because same strand of DNA, but it's, if you look at just the replication fork, it'll look at leading versus lagging and not the actual whole molecule of DNA. So make sure to focus on the fork for the leading and lagging, and the whole, realize that it's the replication bubble. But if one side's leading, the other side's lagging, and lagging, leading. So it's just looking at where it is replicating. So that is the whole thing of DNA replication. And we will get into more specifics of what happens if there's mistakes, and hopefully go over this again. But enjoy.